Good afternoon, everyone.
Hi, good afternoon.
I don't know if anybody told you about Tiffany. She got locked out she's outside her balcony and got locked out in her condo. So she told us to let you know.
Okay, thanks, Yasmin. I guess she has more problems to deal with in microbiology this afternoon. So,
Yeah.
okay, thank you for the heads up. Okay, welcome back, everyone. It's nice to see you, and I do hope you are doing okay. We're going to start rocking and rolling here. Lots to do today. So I'm going to get right back into a conversation with chapter 4. On Tuesday, I sent out your study guide for your first lecture exam. There will be opportunity to discuss that during lab time later today if you have questions, okay? So I will give time for that, okay? So, okay, let's get started here. I'm going to start today by showing you a video, and it's just a video which sort of summarizes some of the things we've already said. But rather than your tired old professor talking to you, you get to see some things visually, and maybe that will help put things into your mind, just help you understand better when you can see it rather than just read pages in a textbook, okay? So we're going to start with a little video here. The video is, what is bacteria? Okay. It's by a graphic illustration company by the name of 3D Animation. I have already placed the link for this video into your notes for today. So if you want to watch it again later during your private study time, you will definitely be able to do that. Okay. All right. So let's just get our juices flowing a little bit here again by watching this introductory video. Okay. Here we go, folks.
Bacteria are single-celled.
Let me get things queued up with good volume for you. Here you go. Okay.
Bacteria are single-celled prokaryotic microorganisms. They can be found everywhere on the planet. The ancestors of modern bacteria appeared on Earth about 3.5 billion years ago. The cytoplasm is the basis of a bacterium cell. It contains the genetic material, that is, the ring-shaped DNA. Unlike in eukaryotes, proteins are not attached to the DNA in prokaryotes. The cytoplasm is enclosed in the cell membrane. All bacteria have a cell wall. It is tough and rigid, and it is made from proteins and polysaccharides. It provides the cells with structural support and protection. Outside the cell wall is the capsule, which is made mainly from polysaccharides. It protects the cell and is responsible for the diseases caused by some bacteria in the host organism. Some bacteria are capable of active locomotion through the use of their flagella. Bacteria have an exceptional ability to adapt and reproduce, and this is why they are extremely widespread. They are capable of both sexual and asexual reproduction. If they reproduce through binary fission, which is a form of asexual reproduction, it results in a large number of individual bacteria cells. Bacteria occur in a wide range of shapes, including spheres, rods, and spirals. Spherical bacteria, which are called cocci, can occur as single cells, pairs, chains, or clusters. Many bacteria that produce suppurative infections or pneumonia are spherical. Gonorrhea is also caused by cocci. One group of rod-shaped bacteria are called bacilli. These can be harmless, such as the lactobacilli used in the food industry. Many bacilli cause diseases, like the pathogens of plague, tuberculosis, salmonellosis, and tetanus. Escherichia coli, which is commonly found in the human intestinal system, is also a bacillus. Syphilis, Lyme disease, and leptospirosis are caused by spiral-shaped bacteria. Gram staining is a method of differentiating bacteria into two groups, i.e., gram-negative and gram-positive. The basis of differentiation is that staining produces different results in the cell walls of different types of bacteria because of their different structure. The two types of bacteria react to antibiotics differently, so treating different bacterial infections requires different antibiotics.
Students with ADHD save 10 hours studying.
Okay, folks. So, that was a fairly simplistic video, but it did do a nice job of sort of re-summarizing the things we were talking about last time. So hopefully, you remember some of those points we were making last time. Do you have any questions at the moment? Okay, please let me know if you do. So what I've done in the notes that we're taking today, the video that I just shared with you is hyperlinked right here in the class notes. Okay, so you can watch it again if you're interested to do so. I've also placed another video link in your notes for a video that I will not show today because it's longer and I don't want to take class time up for the video. But it's a good video about cell biology and the parts of both prokaryotic and eukaryotic cells. So it might be something you choose to watch during your private study time. Okay, so last time we were talking a lot about cell walls. That's where we finished. We were talking about cell walls. We were talking about gram staining. And there's just one more little thing that I want to ask you about or go over before we move beyond the cell wall as a structure that we're describing. Okay, Eric, I see your comment, so we can definitely talk about that later. Victoria, I did get the message. Thank you very much. Okay, so let me ask you in the chat window. Okay, let me ask you in the chat window. Is it gram positive or gram negative that has lipoteichoic acid? Is it gram positive or gram negative bacterial cells that have lipoteichoic acid in their cell wall? Okay, good. It's gram positive. And similarly, I could ask the question, is it gram positive or gram negative that has lipopolysaccharide? And the answer would be gram negative, right? So this brings up the following question. What is meant by the phrase antigenic specificity? What is meant by the phrase antigenic specificity, right? This is a vocab word that was on your exam study guide, okay? This is not something that's terribly well covered in chapter 4 of your textbook, but I want to be sure I cover it with you here, okay? Folks, antigenic specificity sounds like a big, complicated, scary thing, but it's not meant to be, okay? Antigenic specificity refers to the presence of a uniquely identifying molecule. Antigenic specificity refers to the presence of a uniquely identifying molecule. For example, lipoteichoic acid provides antigenic specificity. What does that mean? How should you interpret a statement like this? Lipoteichoic acid provides antigenic specificity for gram-positive cells. Okay. What does this mean? Okay, what this means is the presence of lipoteichoic acid is unique to gram-positive cell walls. You're not going to find lipoteichoic acid on gram-negative cell walls. So think of lipoteichoic acid as a gram-positive cell wall fingerprint. Think of lipoteichoic acid as a gram-positive cell wall fingerprint. It is unique for gram-positive cell walls, okay? This means that gram-positive cells can be uniquely identified by the presence of lipoteichoic acid, okay? Now, and so you could say the following. You could also say the following example. So lipoteichoic acid provides antigenic specificity for gram-positive cells. So let me ask you in the chat window, Here we go. Please give me the complementary statement about antigenic specificity for gram-negative cells. What would be the complementary statement? Okay, I'm just going to go with the first person that wrote because we have lots to do today. Kit, that's a great job. You did wonderful there. LPS, lipopolysaccharide, provides antigenic specificity for gram-negative cells, okay? So in other words, lipopolysaccharide is a fingerprint, a chemical fingerprint that allows a scientist or a motivated person with the right equipment in the lab to identify a gram-negative cell wall, a gram-negative bacterium, okay? Okay. So think of the molecule that conveys antigenic specificity as a fingerprint. Okay, think of the molecule that conveys antigenic specificity as a fingerprint. Your fingerprint conveys your identity, at least to the police department. Okay, so lipoteichoic acid provides antigenic specificity to identify gram-positive cell walls. Lipopolysaccharide provides the same kind of antigenic specificity for gram-negative cell walls. Now, I don't want to get too deep into the weeds right now because we have a lot of things to accomplish. But you might ask, what is the connection between a fingerprint and the phrase antigenic specificity? How, what's the connection here? Okay. Antigenic specificity refers to how animal immune systems are able to uniquely respond to an antigen. Antigenic specificity refers to how animal immune systems are able to uniquely respond to an antigen. So, okay, question in the chat window. Here it comes. If you are infected with a pathogen, what will your immune system likely manufacture in response? Okay, very good, Chelsea and Rachel. Absolutely. Antibodies, right? Antibodies. An antigen is something that's seen as foreign and potentially threatening by the animal immune system. And so what does the animal immune system do? It responds by making antibodies. And what do antibodies do? They bind to specific, unique targets. hence the phrase antigenic specificity. Okay, so this goes back, and I don't know what your biology background is. For folks that have had some AMP, this maybe is a more comfortable topic because you have thought about immunology a little bit, and probably that word antigenic, antigen is not so foreign to you. Okay, an antigen is any molecule that activates the immune system. Okay, typically an immune system an animal immune system will respond to an antigen by making an antibody. Right? Antibodies are proteins that uniquely bind to targets. Okay? So there's a connection here with immunology, is what I'm trying to tell you without getting too thick into the weeds at this point. Okay, let me ask you a question, folks. Are you okay? Is there anything you'd like to ask about this topic? Anything at all? Okay, I appreciate the feedback. Thanks, Elena, Chelsea. Then let's continue. Okay, so let me see what my numbering was here from our last class notes. Okay, so we were looking at the cell wall as being number 2. Now I'm going to go to number 3, which is the plasma membrane. Okay, I'm going to do we're going to continue our tour of a bacterial cell from outside to inside, right? We're getting more internal. We started outside, we're coming inside. All right. Again, you could look at your foundation figure 4.6, foundation figure 4.6 as an overall visual aid today as we're having this conversation. I will also reference other things as we go. Okay, so number 3, the plasma membrane. Okay, true or false in the chat window? A plasma membrane is the same thing as a cell membrane. Okay, folks, generally speaking, that is true. A plasma membrane is not the same thing as a cell wall, but it is the same thing as a cell membrane. Okay, so plasma membrane and cell membrane are synonymous terms. Okay. All right. So, I'm trying to see if I can catch you now. So hopefully I don't catch you on the exam, right? I don't want to catch you on the exam. I want you to do well on the exam. All right. So what can we say about the plasma membrane? You see a diagram of it on figure 4.14, 4.14, which in my book is page 86. Okay. The plasma membrane, also known as the cell membrane, is the boundary of the cell. It is the physical boundary of the cell that separates the inside of the cell from the outside of the cell. Okay, so you would have the outside, you would have glycocalyx, you would have cell wall, you would have plasma membrane. And then you would have the inside, which is where you're going to find the cytoplasm, right? Okay. So let's talk a little bit more about the plasma membrane. You know, what is it made of? Well, you know, the plasma membrane is made of a phospholipid bilayer, 2 rows of phospholipids that are oriented tail to tail, tail to tail. Figure 414, have a look at that. It is made of a phospholipid bilayer with additional molecules sticking through and extending off the internal and external surfaces of the bilayer. Made of a phospholipid bilayer with additional molecules sticking through and extending off the internal and external surfaces of the bilayer. Okay, so let's define what some of these additional molecules are, right? That was kind of a vague, fuzzy phrase here, additional molecules. Well, what additional molecules do we find associated with the phospholipids? Well, we're going to find the following. We're going to find proteins, okay? Just look for yourself in figure 414. Don't you see integral proteins, which are sticking through both phospholipid layers, so they're through the entire bilayer. You see peripheral proteins that are hanging off the inside and hanging off the outside, right? So there's lots of different proteins there. Additionally, you're going to find, okay, so let's see. Yeah, your book is not quite as detailed as sometimes I would like. Let me tell you some other molecules you may find sticking through the bilayer. Okay, you can definitely find some categories of steroid molecules associated with the bilayer. Okay. Steroids, you remember from chapter 2, is a category of lipid. You can sometimes find glycoproteins. These are proteins covalently connected to carbohydrate molecules. You will also find glycolipids. These are lipids covalently attached to carbohydrates. And folks, let me say something that is brand new science, just came out this year, so it's not in your book. Science has discovered that cells can have fragments of RNA sticking on the external surface of the plasma membrane, binding to targets that we don't yet understand. Okay, so small fragments of RNA have been newly discovered on the outside of the cell. Okay, so suffice it to say, everybody thinks that the plasma membrane is just the phospholipid bilayer, and that's certainly a huge important part. I mean, that's like the big honcho, right? But there's all these other varieties of small molecules sticking through the bilayer or hanging off the inside or hanging off the outside that do things that allow the cell to interact not only with the internal environment of the cytoplasm, but the external environment of wherever the cell happens to be floating around. And let's face it, if it's an E. coli cell, the external environment is going to be the inside of your large intestine. Okay, so there's a lot of these things that we're still trying to catalog, identify, and figure out. Okay. The plasma membrane generally is considered the gatekeeper. It's the security guard for what controls the entry of material into the cytoplasm and the exit of material from the cytoplasm, okay? So the plasma membrane controls or regulates the entry and exit of cargo from the cell. Okay. All right. Now let's see here. I'm going to put a statement down and see how you folks wrestle with it. Okay. And so let me just ask some questions here. Okay. Okay. Lance, could you please unmute your microphone and say hi to us?
Hello.
Hi, Lance. How you doing today?
Good.
Okay. Hey, Lance, I got a question for you. Sometimes in books, you will hear the following sort of statement describing a function of the plasma membrane. Here's the statement. The plasma membrane is responsible for catalyzing the breakdown of nutrients. So, Lance, let me ask you, does that mean that the phospholipids are breaking down the nutrients?
No.
Okay, so what does it mean?
That it's recognizing the nutrients.
Okay, what kind of molecule do you think would recognize the nutrient?
I don't know.
So Lance, let me see if I can pull this out of you. For one molecule to recognize another molecule, do you think there has to be sort of a physical interaction between those two molecules?
Yes.
Okay, so would you be okay, Lance, with the idea that there's probably sort of a binding event between these two molecules where they attach to each other?
Yeah.
Okay, so let me phrase this again. What category of molecule is most likely the category of molecule that's going to bind to a nutrient? Let's say, Lance, let's say the nutrient is glucose. Lance, I'm going to give you a list of possible answers and see if you can get it. So I'm fishing for whether the molecule is a carbohydrate, a lipid, a protein, or a nucleic acid.
I'm not sure.
You got 25% chance of getting it right, Lance. Give it the old college try.
What were my options?
Okay. The options are carbohydrate, lipid, protein, nucleic acid.
Lipids.
Okay, thanks for trying, Lance. But thanks for participating. I do appreciate it. I'm fishing for protein. And what I'm trying to say is, okay, when you read books that say that the plasma membrane catalyzes the breakdown of nutrients, that is true, but you need to interpret it correctly. This means that there is an enzyme. There is an enzyme embedded in the phospholipid bilayer that binds the nutrient and breaks it down. Okay. If you hear the word catalyze, you should be thinking about enzymes, not phospholipids. Phospholipids do not behave as enzymes. Carbohydrates do not behave as enzymes. Lipids do not behave as enzymes. Proteins definitely behave as enzymes. And generally speaking, nucleic acids don't, but there is an exception there. There are some catalytic pieces of RNA that have been discovered. Okay. So if you had told me RNA, I kind of would have had to give you that, okay? But thanks, Lance, for participating. I appreciate it, really do. Chelsea has a question in the chat window. Is this why it, and I'm assuming it means the plasma membrane, is considered semi-permeable because it has to break down the two large proteins? Well, I could say yes to your question, Chelsea. But you know, for the example, when I was trying to get Lance to say something about glucose, right? Even glucose is too large necessarily to get through the plasma membrane without assistance. So it's not necessarily large proteins, but it's any large cargo might have a really hard time getting through the plasma membrane. Okay, any other questions? So I don't want you to think that when you say the plasma membrane breaks down the nutrients, that it's the phospholipids. It's not. It is the enzymes, the proteins embedded in the phospholipids that do the work. Okay, same thing with this next phrase. Okay, the plasma membrane manufactures energy molecules called ATP. Now remember, gang, everything I'm telling you here in chapter 4, I am customizing it for a conversation about prokaryotic cell biology. Prokaryotic cell biology. So for you experts on animal cells from AMP, if I said that an animal cell plasma membrane made ATP, you would be like, huh? That's not right, because we all know, type into the chat window, where do animal cells make ATP? Okay, good job, everybody. It is predominantly the mitochondria. So, right, the mitochondria in animal cells, in eukaryotic cells, makes ATP. But in bacteria, it's not the mitochondria, it's the plasma membrane. And again, I won't take time to do this. It's not the phospholipids that's making the ATP. It is not the phospholipids, it's the enzymes that are the proteins embedded in the bilayer that's making the ATP. So just trying to get you to contextualize how this information needs to be processed. Any other questions? Okay. All right, gang. So while we are here with the plasma membrane, this seems like quite a nice time to talk about methods of moving cargo through the membrane, either into the cytoplasm or out of the cell. So we're going to take a slight detour at this time and talk about moving cargo through the plasma membrane. Okay. So yes, I'm talking about number 3 here. Plasma membrane is a structure. We're going to make, well, it will make letter E a conversation about transport through the plasma membrane. And I'll underline it because this is a pretty significant topic that we need to talk about. Okay. So, all right. When you talk about transport through the plasma membrane, there are going to be passive processes and active processes. And so hopefully you understand the difference there, right? A passive process takes place without the cell expending its own energy, without the cell burning ATP. So maybe I'll just put that down there. Passive process, no ATP required. And active processes, ATP is required. Okay, so let's list out some of the passive processes. Passive processes would include simple diffusion, facilitated diffusion, active transport, and osmosis. Now, truth be told, I kind of already expect you know these things. I hope you know these things, right? I don't want to have to recover. I don't want to have to cover these things again. But at this, let me just pause and ask you, do you need me to explain any of these processes to you? Simple diffusion, facilitated diffusion, active transport, osmosis. Do you need a little refresher on any of these terms?
Can you do facilitated diffusion?
Okay, all right. Thank you, Samantha, for being brave and asking, because that's what I want from you. Otherwise, I will move on. Okay, so Samantha has asked about facilitated diffusion. So facilitated diffusion is the movement of a solute across the plasma membrane from an area of high to low concentration with the help of a carrier protein. Okay, with the help of a carrier protein. Let me read this again. Facilitated diffusion is the movement of a solute across the plasma membrane from an area of high to low concentration with the help of a carrier protein. In other words, there is a protein embedded in the plasma membrane. Take a look at figure 417. Take a look at figure 417. Okay. Take a look at figure 417. Samantha, please unmute your microphone and tell me what color the transport protein is in figure 417, picture C.
It's purple.
Thanks, Samantha. I just wanted to know you were looking at the right spot. You're absolutely correct.
Thank you.
Okay. All right. So, the example your book gives you there, it's showing you that glucose would be an example of a solute that's going to require this carrier protein to move the cargo through the membrane. You're always going to move it across the membrane from wherever it is most highly concentrated to where it's most lowly concentrated. So glucose is more highly concentrated in the cytoplasm, then the glucose is going to leave the cell. If the glucose is more highly concentrated outside the cell, then the glucose is going to enter the cell by facilitated diffusion. Okay, there was also a request in the chat window for osmosis. Okay, osmosis is, figure 418. Okay, and so you do need to be aware of this. You do need to think about this, right? It is going to be an essay question for you on your exam. Osmosis is the movement of water across a membrane from an area of high free water concentration to an area of low free water concentration. Okay, so Chelsea, thank you. Yes, I do have a typo in my notes, don't I? Active transport should not be there for passive processes. Thank you, Chelsea. I got carried away. Okay. So facilitated diffusion osmosis. Osmosis is the movement of water across a membrane from an area of high free water concentration to an area of low free water concentration. Aubriana, please unmute your microphone and say hi to us.
Hi.
Hi, Aubriana. How you doing this afternoon?
Good.
So, Aubriana, I want to tickle you a little bit on this topic of osmosis. What component of a solution has the ability to lower the free water concentration of the environment?
Component.
Right.
The solute, the being a solute.
Good, good. Stay with me, Aubriana. You're absolutely right. What does the phrase free water mean?
Free water.
Yeah, what does that mean?
I'm not sure.
Okay. Thanks, Aubriana. Let me just put this down in your notes. Free water means that the water is not interacting with the solute. Okay. Let's see. Hey, Kit, could you please unmute your microphone and say hi to us?
Hello.
Kit, how you doing today?
I'm hanging in there.
Okay, good. I hope you are. Hey, Kit, could you tell us what property of water causes it to interact with most solutes?
And the fact that it's polar.
Beautiful answer. Thank you so much. You did a great job. Remember, water is a polar molecule, so it has a positive end, right? The hydrogen atoms, and it has a negative end, the oxygen atom. Because water is polar, it tends to interact with solute. If it interacts with solute, the water is no longer free. It is no longer free. Okay. Water is polar. Okay, folks, let me say on YouTube, if you go to YouTube and you do a search for the following phrase, how osmosis works, that will give you a quick little 2-minute video on osmosis. I think it might be helpful for some of you to review that. Okay. Aubriana, is any more aspect of osmosis that you were hoping that I would talk about?
No, I think that's okay.
Okay. Obviously, I didn't completely answer the topic for you because I could do a half-an-hour lecture on osmosis, and we just don't have the time. But if you want to stick around after class today, after lab, I know office hours before the exam, I'd be happy to help you, okay?
Okay, thank you.
All right. Okay, so, excuse me. Okay. Passive processes, simple diffusion, facilitated diffusion, and osmosis. You really should know them. You really should know them, okay? I'm counting on that, the fact that you've had some exposure to this content previously. Then we have the active processes, okay? And this deserves some attention as well. By active processes, I'm going to say active transport, active transport, and active transport is going to include something that is unique to bacterial cells. Animal cells don't do it. Animal cells don't do it. Generally, we don't think eukaryotic cells do this. Something unique to prokaryotic cells. It is a process known as group translocation. I don't have a figure number to refer to because there is no figure. But under the heading of active processes, which in my textbook is page 89, there's a bold-faced term called group translocation. Okay, so active transport is the reverse of facilitated diffusion. Active transport is facilitated diffusion, except you move the cargo from low to high concentration. Okay. Group translocation is a subtype, a subcategory of active transport. Okay. This is a type of active transport thought only to be performed by prokaryotic cells, in which the cargo being moved is modified or changed during transport. Okay, let me give you a silly analogy. That would be like you ordering a television set from Best Buy, and the people at the Best Buy store put your television set in the delivery truck, and while the delivery people are driving the television set to your home to deliver it, the television set is changed to become a laptop computer. Okay, so that tells you something. Most of the transport processes by far that we talk about is simply moving the cargo from point A to point B, and there's no modification in the cargo itself. The cargo is never usually changed in any way. But with group translocation, the cargo is changed en route between its starting point and its destination point. This is unique as far as we can tell to prokaryotic cells. Okay. All right. So I will also say here, for the benefit of my A&P friends, that when we talked about active processes in A&P, we talked about things like endocytosis and exocytosis, right? Phagocytosis, all those beautiful words. Those processes do not occur in prokaryotic cells. Prokaryotes do not perform exocytosis or endocytosis. Okay, so probably one of the challenges is many of you have some pretty good biology background knowledge to draw upon, but now you need to begin to compartmentalize what is true for a eukaryote versus a prokaryote. Okay. All right. Folks, do you have any questions? Okay. I know I'm going fast, so thank you for hanging in there. All right, so number 3 was plasma membrane. We're now going to continue our tour here of a typical prokaryotic cell. Number 4 is the cytoplasm. I'm not going to say much about it. The cytoplasm is the inside of the cell. The composition of the cytoplasm is kind of jelly-like or honey-like or molasses-like, which is to say it's not just water, but it's water with a variety of dissolved solutes in it, which changes the physical characteristic of the water. So it's more like a jelly, like strawberry jelly, grape jelly, that kind of thing. Okay. This is the internal fluid of the cell, which is made of water plus a variety of solutes and has a jelly-like composition. Okay. So if you were to puncture a cell, like if you had a tiny little needle and punctured a cell, you would not see water gushing out of it as though you popped a balloon. That wouldn't happen. what would happen is that a slow sort of moving jello-like substance would start to ooze out of the puncture, okay? All right. Now let's get to some good stuff here. Okay. Nucleoid region. Nucleoid region. Okay, the nucleoid region is shown to you probably best on the foundation figure, foundation figure 4.6. Okay, this is the region of the bacterial cell that contains the bacterial chromosome. Okay, the nucleoid region is not a nucleus. It is not a nucleus because there's no membrane there. There's no membrane around the chromosome. And that's one of the gotchas, right? Somebody on the exam is going to tell me nucleus rather than nucleoid. And while most people will say, well, I don't care, big deal, splitting hairs, here in micro, we care about these details. The nucleoid region is not the same thing as a nucleus. There is no membrane, okay? Some things that I would ask you to be aware of here, okay? Talking points about the nucleoid region. Typically, most prokaryotic cells that have been identified and studied by science, by far, most prokaryotic cells tend to have one single circular chromosome. You know, contrast that with what you know to be true of your chromosomes in your cells, where you have 46 linear chromosomes that when they're getting ready to reproduce through mitosis look like little X's, right? Bacteria's chromosomes don't look like that. They look like circles. Now, if you look at the foundation figure, 4.6, which in my textbook is page 76, when you look at where the chromosome is, the nucleoid containing DNA, it's got a purple color, right? It really doesn't look like a circle, does it? Give me some descriptive language in the chat window. What does the chromosome look like to you with the foundation figure? What does the chromosome look to you? What does it look like in the foundation figure? Okay, it looks knotted, curled, rubber band, noodles or worms, spaghetti, beautiful, good stuff, okay? I tend to say it kind of looks like purple yarn, right? Purple yarn, it's all jumbled up, something like that. That's my point. Now, here's the deal. If you took a purple rubber band that was completely circular, and you took one side of the rubber band and stretched it across the other side, and kept repeating that process till you stretch one side across the other side, all of a sudden you would get a structure that didn't look circular because it was folded up on top of itself. And that's what's happening here. If you were to unravel the purple yarn of the foundation figure, it would unravel into a shape which is roughly a geometric circle, okay? Next. The video I shared with you today, I like the video, but there was one thing on it I did not agree with. The video told you that prokaryotic DNA is not associated with protein. That's really not true, okay? What I will say is this, prokaryotic chromosomes are associated with proteins called non-histone proteins, okay? if you have any animal biology background, you know that the chromosomes in your cells are made of DNA plus proteins called histones. Bacterial cells do not have histones, but they do have other proteins which are generically referred to as non-histone proteins. Okay. All right, prokaryotic chromosomes, such as you see in Foundation Figure 4.6, are often physically tethered to the inner surface of the plasma membrane. They are connected, they are attached to the inner surface of the plasma membrane, even though it's not drawn that way in the Foundation figure. Okay. Even though it's not drawn that way in the Foundation figure, it is nonetheless true. Okay. And scientists think that it is attached to the plasma membrane to support cell reproduction processes that you may know are called binary fission. Long story short, a very interesting report came out in the scientific literature this month revealing that our own DNA in our own human cells appears also to be connected to the inner surface of the nuclear membrane. That was a shock to many scientists that we hadn't discovered that before, but it seems to be the case, which makes you wonder about the evolutionary relatedness between bacteria and higher eukaryotic cells. All right, let me give you an idea about size. Okay, so, right, size. Let's do this. Human cells have 46 chromosomes with 6.4 billion nucleotide pairs of DNA. A typical E. coli bacterial cell has one chromosome with 4.6 million nucleotide pairs of DNA. Okay. So the point I'm trying to make is if you look at the scale of things, right? 6.4 billion for humans, 4.6 million for bacteria. So yes, you might say that bacteria are simpler organisms. I don't think we're ever going to find an E. coli cell that's writing an English novel. That's probably true. However, don't forget, E. coli and other bacteria often have different genes than animal cells, and these different genes allow the bacterial cells to perform chemical reactions our own cells cannot perform, such as manufacturing vitamin K. So E. coli might be simpler, but it doesn't mean they're not capable. They're actually quite capable of doing things that our own cells are not capable of doing. Okay. Thoughts, questions, cuss words? All right, let's keep going. Number 6. Okay. Number 6, the video pointed out, and the Foundation figure on Foundation figure 4.6, again, page 76 in my book. It shows you something called the plasmid. Okay. Plasmids are extra chromosomal pieces of DNA. Okay, and let me emphasize that. Extra chromosomal pieces of DNA. Okay. If you look at the Foundation figure, you can see in the bottom right corner inside the cell, there's a small purple rubber band called a plasmid. You can see it is not physically part of the chromosome. It's not physically part of that purple yarn. So we describe plasmids as extra chromosomal pieces of DNA. Let me tell you that by far, most eukaryotic cells do not have plasmids. This is a prokaryotic entity, okay? Most eukaryotic cells do not have plasmids. This is generally a molecule found only in prokaryotes. Okay, I will say there are some rather strange looking molecules of DNA that exist in some weird types of fungi that have a plasmid-like appearance to them. And fungi, of course, are eukaryotes. Okay, one notable exception would be fungi. Okay, all right. So what should we tell you about the plasmids? Well, compared to the giant size of both animal cell and bacterial cell chromosomes, plasmids are very tiny. The typical size of a plasmid is about 2,000 to 50,000 nucleotide pairs of DNA. Okay, so the human genome has billions of nucleotides of DNA. The E. coli chromosome has millions of nucleotides of DNA. And E. coli plasmid is going to have a few thousands of nucleotide pairs of DNA. Plasmids are pretty neat. Let's say a few things about these things. Plasmids often contain genes that are not present on the chromosomes. Often contain genes not found on the chromosomes. And these genes often convey a survivability advantage to the microbe. These genes on plasmids often convey a survival advantage to the microbe. For example, okay, we all know, many of you know, because you're going into medicine, you may already work in the healthcare field, that we have an ever-present problem with antibiotic resistance, and that's a whole nother conversation for another day. But the point of it is the bacteria that are resistant to the antibiotics, 99 times out of 100, the genes inside them that allow them to resist the antibiotic and not die because of the antibiotic are genes that are found on plasmids. Antibiotic resistance genes are found on plasmids. Okay. So Kid has a comment on in the chat window. So that's fun. There's a video game called Bioshock where there are plasmids that are basically serums that you can use to manipulate your body to have certain abilities. Absolutely, right? I mean, guess what? And I say this tongue in cheek, but not entirely. I could cry when I say this. Scientists don't make a lot of money. So they moonlight as expert consultants for video game designers, okay? I'm aware of Bioshock, Kit. I've never had the pleasure of playing it, but I'm aware of it. So thanks for saying that. Okay. Plasmids are small enough that they can be routinely exchanged between different bacteria. bacteria can actually swap plasmids in sort of a conjugal way, kind of a bacterial sex method that can happen. Okay. Now, gang, I'm going to say something next that might be mildly disturbing. Okay. At the animal level that you and I live our daily lives on, right? Human beings, or talking about dogs or cats or giraffes or polar bears or whatever it might be, right? There are reproductive barriers in place that Mother Nature has placed so that, different species of animals generally cannot crossbreed and reproduce with each other for the most part. Okay, what I'm saying is you cannot crossbreed a polar bear and a giraffe and get some kind of hybrid animal out of it. That just don't work because of reproductive barriers that Mother Nature has put into place. Those reproductive barriers are much less present in the microbial world, which is why an E. coli bacterial cell can kind of have the equivalent of bacterial sex with a Vibrio cholera bacterial cell and exchange plasmids. What I'm saying is that one of the driving forces of evolution on our planet is the free roaming exchange of plasmids and other small genetic elements at the microbial level, where there is no reproductive barriers against the swapping of genetic material from one species of cell to another species of cell. Okay, so it drives evolution, but it's also problematic, right? Because if you're infected with a certain kind of bacterium that's antibiotic resistant, it can go off in the environment and mate with a different species of bacterium, swap the plasmid over, and all of a sudden the second bacterium is now also resistant to antibiotics, and it's a problem. It's a problem for medicine in the healthcare community. Okay. All right, so there's a lot of neat things that could be said about this. We could do a whole course on plasmids, believe it or not, but I'm going to just say one more thing. Because plasmids are tiny, it's convenient to manipulate them in the modern biology lab. Plasmids are often one of the workhorse vehicles for genetic engineering. Plasmids are commonly used as a piece of technology for modern genetic engineering. Okay, so let me ask you this in the chat window. Do you know anyone that takes insulin? Okay, this keeps coming up, doesn't it? Diabetes and all this kind of stuff. Okay, folks, that insulin that person is taking in the United States of America, it was made inside a bacterial cell. And it was made by having a gene that tells the cell how to make the insulin protein present on a plasmid that was genetically engineered into the bacterial cell. So what I'm saying is many of our medicines today and things that have been around a long time that you know of as insulin is now manufactured as a biological product through using plasmids in the modern pharmaceutical and biotechnology industries. Okay. Insulin is now made inside bacteria from genes on plasmids. Okay. All right. Thoughts, questions? Okay. Okay, let's keep rolling here. We got about 5 minutes before we got a break. Okay. Number 7 is ribosomes. Okay. Ribosomes, as you know, are nanomachines that manufacture protein. They're nanomachines that manufacture protein. I'm just going to give you some talking points here about these guys. Ribosomes, you know, the average number of ribosomes in a typical E. coli bacterial cell will be around 50 to 60,000. 50 to 60,000 ribosomes in one bacterial cell. That's in contrast to a typical human cell that might have hundreds of thousands of ribosomes, okay? So a typical bacterial cell will have 50 to 60,000 ribosomes. The actual number of ribosomes varies from one moment in the cell's life to another based on the environmental conditions the cell finds itself in and what the cell is trying to accomplish. Is the cell just trying to live, or is it trying to reproduce? That makes a huge difference. Okay. As we've learned before, bacterial ribosomes are described as 70S particles. Okay, 70S particles. And you do see a little bit of graphical information trying to describe that to you on figure 419. Figure 419. In my book, it's page 91. Okay, so I don't know if in your geekiest dreams you've ever wondered what a ribosome itself is made up of, but a ribosome itself is made of its own proteins and own ribosomal RNA. When you say ribosomal proteins, that's often abbreviated R-protein for ribosomal protein, and ribosomal RNA, which is little r RNA. Okay, that's what these machines are made out of. Okay. Ribosomes, we said probably last class period that the cell wall of bacteria have been a traditional target of antibiotics, such as penicillin, which disrupts the cell wall synthesis of many species of bacteria. Ribosomes have also been a classical target of different antibiotics. For example, streptomycin. For example, streptomycin is an antibiotic that disrupts bacterial ribosomes. You'll learn more about that in chapter 20 when we get to it. Okay. All right. Sorry that I'm going so fast, folks. There's just so much to do here. Okay. Do you have any questions right now? Okay. I don't think it makes sense to continue with the conversation with only like 50 seconds left here. So we're going to pick up this conversation in lab today. Expect at least a half an hour, maybe more of additional lecture. And we'll have some other things to share with you in lab, okay? So do you have any questions before we pause? We'll take a 15-minute break and start back up at 2 o'clock. Hi, folks. Welcome back. If I could draw your attention back to our common space here, we're going to get started with our lab time, which is to say I'm going to continue with some lecturing from chapter 4, okay? All right. So we're going fast here today, covering a lot of content. Do you have questions you'd like to ask at the moment?
I just got my book in the mail, so I'm kind of looking everything over now because I wasn't able to before because I was having issues with Red Shelf. So I just want to make sure the test that we're gonna have is on 1, 2, 3, and 4.
No, Tiffany. It is on 1, 2, and 4.
Okay, that way I can review everything and look it all over this weekend.
Yeah, chapter 3 is not on the first exam. All right, so let me, I want to bring a question poll up for everybody to give an answer on, okay? So hold on a second. Here comes your poll. Please give me your response. Okay, so just giving everybody a moment to actually vote on things, okay? Okay. All right, so I'm going to close the poll in about 10 seconds. Okay, so I'm going to end the poll and I will share the results with you. Okay, you can see that the majority vote on this poll was to have the exam on Thursday. So here's the deal, folks. I know I'm moving quickly, so I'm willing to consider moving the exam to next Thursday. There's a couple of things you have to understand, however. Even if we move the exam to next Thursday, you will still have a lab assignment due on that same day. I won't change the due date for the lab assignment. Also, if we move the exam till next Thursday, I still intend to start lecturing from chapter 8 on Tuesday. Okay, I don't have the luxury of slowing down. I can try to give you a little bit longer to prepare for the exam, but I need to keep moving with the curriculum.
So will we have lecture on Thursday for the exam? I mean, in like swapping it, like Tuesday, we normally would take the test and no exam. I mean, take the exam and no lecture.
So Elena, just to clarify for you, if we move the exam to Thursday, then we would have normal class on Tuesday. I would lecture on Tuesday. Thursday, during our lecture time, you would take your exam. And then after lecture, we would have our normal lab period.
Okay, I understand that. I'm familiar with that. I just wanted to clarify
Sure.
that that's the way you would continue doing it.
Okay.
Can we redo the poll now with that information?
Hold on a second. Hold on a second.
I still think it would work though, because the only thing you're doing is switching when you're going to lecture on chapter 8. You're just moving it to Tuesday instead of Thursday, right?
Yeah, but we're simply exchanging when I give you the lecture for chapter 8. That's all we're doing. Okay. Hold on a second. Okay, technology. Okay. Here we go. now what day do you want your exam to be on, Tuesday or Thursday? Okay, we'll close the poll in 10 seconds. 5, 4, 3, 2, 1. Here we go. Here's the results. You can see that Thursday is still the preferred day. Okay, gang, that's okay with me. So we'll move the exam to next Thursday, so a week from today. Okay, with the understanding that you still will have a lab activity, lab homework that will still be due on that day. And on Tuesday, I will lecture from chapter 8. Okay. So maybe that gives you a little bit more time to prepare, trading one thing for the other thing. Okay. So, all right, we'll do it on Thursday. And when I send out this afternoon's email, I will take note that the exam will be next Thursday. Okay. All right. Any other thoughts or questions before we continue with the lecture? Okay. Let's continue then. All right. Okay, so we were talking about ribosomes. Bacterial ribosomes are described as 70 S particles. I want to make the point that eukaryotic ribosomes are 80 S. If you don't know what the S stands for, it is described in your book. It's a Svedberg unit. It's a unit which refers to the sedimentation rate of spinning cells through a centrifuge. Okay. And if you're really curious, I can describe it to you, but it would take me a while to do that. So I'll move on for now. And if you want to ask a question about it, please do. Okay. All right. Number 8. Okay, so I'm just hitting the highlights here of major topics that I want you to be familiar with. The next topic that I'm going to talk about is actually the endospores. You see a picture of endospore formation on figure 4.21. Figure 4.21. Okay, so let me define it, then I'll give you some talking points. Endospores are extremely durable, tough, dehydrated cells that actually live inside other cells. I mean, they are created inside other cells, and then the mother cell will die away, and you will just have this very tough, dehydrated cell that's almost in a hibernation state, kind of like a hibernating bear. Okay, so these are extremely durable, dehydrated cells that form within a quote-unquote mother cell and eventually may exist on their own outside the mother cell. Okay, let me give you some talking points here. You know, if we were together in lab, one of the things we do is we actually prepare an endospore stain and get you a chance to look at it under the microscope. Unfortunately, we won't have that experience. Let me see if I can bring up an endospore stain in Google Images really quick for you. Yeah, something like this here on the upper right. Okay, all the cells that kind of look like a bluish green, those are endospores. All the cells that are still pinkish and rod-shaped, right, bacillus, those would be the mother cell that formed the endospores. Okay, All right, so talking points about endospores. Endospores are only formed inside and by gram-positive cells. Only gram-positive cells can make endospores. Okay, gram-negative cells do not have the genetic instructions to do it, so they don't. Endospores are formed when the mother cell, in the picture I just showed you, those pinkish red cells, endospores are formed anytime the mother cell feels threatened by its environment. Okay, so formed by a mother cell when the environment is threatening to the mother cell. So that would mean that if the environment lacks the necessary water for the mother cell to live, the mother cell would try to form the endospore so that when the mother cell dies, the species of cell can still continue on through the endospore. If the environment has some kind of toxic chemicals in, maybe some heavy metals, maybe it's overexposed to too much ultraviolet radiation from the sun, a variety of different things that the mother cell might feel threatened by, mother cell will attempt to build an endospore before the mother cell dies from the environmental circumstances. Okay, an endospore can survive very challenging conditions. Okay, once formed, the endospore can survive an environment where there's a lack of water, can survive with a lack of water, it can survive extreme heat. And by extreme heat, I mean even up to the boiling point of water. Okay. Some species of gram-positives will form endospores that are capable of surviving even a few minutes of boiling water. That's how hot they can survive in. The endospore forms by layering itself with many thick coats of peptidoglycan, right? We talked about peptidoglycan before. So if you make more and more layers of peptidoglycan, and you basically form a peptidoglycan shell, which is largely impermeable to many toxic chemicals from the environment that might want to get into the cell. So endospores can survive the presence of toxic chemicals and even honestly moderate exposure to various forms of radiation. Certainly endospores could survive a level of radiation exposure that would quickly kill a human being. Okay. So let me mention this here, right? The endospore forms by building a multi-layered structure of peptidoglycan around the genetic material. Kit asks, are they the tardigrades of the prokaryotic cell world? I think that would be an appropriate analogy, Kit. Yes, they are pretty tough. Okay. Obviously, endospores do present a problem for humanity in certain circumstances, right? Think about the food industry, food spoilage, the canning industry where you're trying to preserve foods. The last thing you would want to do is go into your grandmother or grandfather's house and get some canned food that they prepared 40 years ago and open it up and eat it, right? I mean, God bless you. You love grandma and grandpa, but that would not be a safe thing to do because there's a very good chance there's going to be endospores and other nasty things in that that can, okay? So endospores are a concern for the food industry and canning industry. Obviously, those are related. Endospores can also be a broader concern to the medical community. Generally speaking, most microbes that are inside your body, well, they're not going to form an endospore because they're living off nutrients in your body and they're happy to do so. However, if you are exposing your body to toxic chemicals or radiation, let's say for cancer treatment, which could also harm these microorganisms, then in some cases, some species of microbes might start to form endospores inside your body. Okay, so it is a concern for the food industry and medicine. Endospores that somehow, through whatever procedure, end up on surgical equipment, knives, scalpels, things like this, they're really hard to kill, really hard to kill. Okay, and that's why, at least in most hospitals in the United States, you know, when you go in for surgery, they're definitely going to use brand new surgical tools during your surgery. They're not reused, they're not clean, they're not sterilized, they're brand new. But that's not true all over the face of the planet, right? Particularly in poor nations, third world nations, they will try to reuse surgical tools, scalpels, things like this by trying to clean them in between surgeries. And they have a lot more trouble with post-operative infection because of this. Okay, so endospores are concerned for the food and canning industry, as well as for modern medicine. Okay, thoughts, questions? All right, so folks, that's a pretty good rundown of prokaryotic cell biology. I've tried to point out things that are important for you practically on your exam, and also make a point of distinguishing topics, distinguishing things that make these cells different from a typical animal cell, like our own cells in our bodies. Okay. Now, this whole chapter 4 that you're going to be taking an exam on is functional anatomy of prokaryotic and eukaryotic cells, right? That's the title of it. And so some principles we've talked about are certainly as true for prokaryotes as they are for eukaryotes. Osmosis works the same, whether it's a prokaryote or eukaryote, it's the same thing, right? Same thing. Okay, I've spent most of my time talking about prokaryotic biology because this is a micro course. You will be expected to know some basic ideas about eukaryotic cell biology. And so what I'm just going to do is create a laundry list of structures that I think you should know for your exam under eukaryotic cell biology. I'm not going to lecture on the structures. I'm going to leave it to you to do that. Okay. Eukaryotic cell structures, important to you. Okay. So here's a quick list of these things. And you're going to see that some of these things are certainly in common with prokaryotic cells. I mean, the details might be different, but the overall idea is the same, right? Plasma membrane, cytoplasm, cytoskeleton. You should certainly know the nucleus, which you know is not the same thing as the nucleoid region. You should know the endoplasmic reticulum. Endoplasmic reticulum is not found in bacteria. We didn't talk about it, right? So endoplasmic reticulum, both the smooth and rough varieties. Golgi complex. Mitochondria. Ribosomes. Lysosomes. Centrioles. Okay. I think that's a pretty good starting point for having some basic knowledge of eukaryotic cell biology. And I'm hopeful that at least for many of you, those terms are familiar. Okay, this is Microbiology 221. That means this is a sophomore level bio course. So I expect that you have some background that you can quickly get up to speed on. Okay. Okay. Do you have questions right now? Okay, there's one more thing I want to talk to you about today, and that is the theory that science most uses to explain how eukaryotic cells came to be. Okay. The video I showed you today at the beginning of class said that the most primitive forms of prokaryotic life probably emerged on the planet about 3.5 billion years ago. Depending on whose data you want to argue, the first primitive eukaryotic cells probably came into existence about 3 billion to 2.5 billion years ago. Obviously, this is phenomenal expanses of time that we can't really fathom because we only get to live 100 years on the planet if we're lucky. Okay, but let's talk a little bit about the endosymbiotic theory. Okay, this is a theory which attempts to explain the origin of eukaryotic cells from prokaryotic cells. Okay. Nobody, I don't care who it is, if they say, I know how a prokaryotic cell evolved on the face of the planet, they're lying to you. Nobody knows that. Okay, we have some guesses. Now, we know about lipid bubbles forming in the early primitive atmosphere of the planet and the oceans. We know about virus-like genetic elements known as transposons that were probably around a long time ago. But, let's not kid each other. It's not like we can go back in a time machine and watch this happen. So we don't really know where and how the first cells developed. We just don't. Okay. But we do have some ideas about how the first primitive cells, which are prokaryotic, eventually became eukaryotic. And the basic idea is probably the basic idea is simple, although the details of how this might have actually happened at the biochemical level are still kind of muddy. Here's the basic idea. Okay. What this theory proposes is that at some point in the evolutionary history of cells on planet Earth, there were large prokaryotic cells and there were small prokaryotic cells. And through conditions which we may not understand, it seems to be the case that sometimes a small prokaryotic cell would bind to, merge, and fuse with a larger cell and internalize itself into the larger prokaryotic cell. Well, if that's true, then that would suggest that at least some of our present organelles in eukaryotic cells are actually symbiotic bacteria that live inside our eukaryotic cells. That's what the theory is proposing, okay? Basic idea is that small prokaryotic cells entered large prokaryotic cells and came to exist in a mutually beneficial relationship with each other. It's kind of like, the analogy would kind of be like two soap bubbles in your kitchen sink fusing together. And if you've ever washed dishes, if you've ever washed dishes and you just take a moment to watch the soap bubbles kind of merge and fuse and then sometimes separate, depending on what's going on, that's the basic idea here. Kit, your question, is that where the idea that the mitochondria is a bacterium came from? That's one piece of information, yes. So the fusing of soap bubbles in your kitchen sink. Now, obviously, the last time I saw soap bubbles merge in the kitchen sink, life did not come out of that, okay? But this is the idea that science is proposing. So what is the evidence? Let's go over the evidence of why we think this might be true. What is the evidence? Okay, here's the evidence. Okay, and we'll start with Kit's statement there. Okay, scientists have discovered over the years that eukaryotic cells, like the cells that make up our bodies, they have at least two sources of DNA. They have the DNA in the nucleus, and if it's an animal cell, they also have DNA in the mitochondria. Mitochondria has its own DNA. Okay, I don't know if that's new to you or not, but it is nonetheless true. Mitochondria in animal and plant cells has its own DNA. And for plant cells, which obviously have chloroplasts, chloroplasts have their own DNA. Now, obviously, animal cells don't have chloroplasts, but plant cells have chloroplasts to support photosynthesis, and those chloroplasts, organelles, do have their own DNA. So I'll just stick with the animal cell right now. Mitochondria has its own DNA, okay? If you take, with modern DNA sequencing technology, if you take and you sequence the nucleotide sequence of the mitochondrial DNA, the A's, T's, C's, and G's, which we will talk about in depth in chapter 8, when you sequence the nucleotides of DNA that come out of the mitochondria, and then using a computer, you compare the sequence of DNA that comes out of a mitochondria to the rest of the DNA you find in the nucleus of a typical animal cell, as well as the typical DNA you find in the nucleoid region of a bacterial cell, you can guess the punchline. The mitochondria DNA is much more similar to the nucleoid DNA of the bacterium than it is the nuclear DNA of the animal cell. Okay. Sequence comparison of mitochondrial DNA reveals DNA that is more similar to bacterial DNA than animal cell, I'll just say eukaryotic cell DNA. That's kind of curious, isn't it? I mean, that's just kind of odd that this observation seems to be true. Okay, well, let's take a look at some other things. Let me ask you this in the chat window. Let me ask you this in the chat window. Where inside your cells will I find ribosomes? Okay, good stuff. Thanks for those that are typing things in there. I really do appreciate that. Okay, and that's that's wonderful. Right, the endoplasmic reticulum, or as Rachel said, what they said. You read the endoplasmic reticulum, the rough ER. There are some free-floating ribosomes in the cytoplasm as well. But folks, did you know that mitochondria have their own ribosomes? If you purify mitochondria out of a cell and then open up the mitochondria and look inside, not only are you going to find DNA, you're going to find ribosomes. Mitochondria have their own ribosomes. Well, that's kind of weird, right? I mean, why would an organelle that makes ATP for the animal cell have its own ribosomes? That seems strange. Okay. Well, long story short, if you do a meticulous analysis of the structure and composition of a mitochondrial ribosome, and you compare it to the structure and composition of normal bacterial ribosomes or ribosomes we typically find associated with the rough ER in an animal cell, you can guess the result. Mitochondrial ribosomes are much more similar to the bacterial counterpart than the animal cell counterpart. Comparison of mitochondrial ribosomes reveals that mitochondrial ribosomes are more similar to bacterial ribosomes than the ER ribosomes of eukaryotic cells. All right. Well, that seems just too odd to have those two pieces of information together. Let me share another one with you. Here's the third piece of evidence. Okay. Question in the chat window. What are the names of the cell reproduction processes that animal cells use to copy themselves? Okay, so Kit happens to be first off the gate today. Taylor, thanks so much. Okay. I think Kit has his fingertips glued to his keyboard, right? It's just boom, right there it is, okay? So mitosis and meiosis. So here's the deal. When bacteria reproduce, they use a process known as binary fission, which we haven't talked really about at all yet. We will later. Mitochondria happen to have the capability to reproduce independently of the rest of the cell cycle of a eukaryotic cell. Mitochondria can reproduce even when the animal cell is not reproducing. Mitochondria can reproduce when the animal cell is not going through mitosis and meiosis. And if you study the process of mitochondrial reproduction, guess what? It very much resembles binary fission. It does not resemble mitosis or meiosis at all. That's weird, gang. That's very weird. Okay. Mitochondria reproduce in a process similar to binary fission. Okay. They can reproduce independently of mitosis and meiosis happening inside animal cells. Okay, so if I think hard enough, maybe I could come up with another piece of evidence. I'm sure there is, but long story short, there seems to be evidence that the mitochondria, the chloroplast, maybe the nucleus represent some form of primitive symbiotic prokaryote that merged into a larger cell a long time ago and became the current organelles they are in modern eukaryotic cells like our own. Okay, now does this prove it? No, we can't say it's definitely 100% true, but it seems weird that this, that we can make these measurements and come to these observations. So let me just ask you, and you can give me an honest answer. Have I convinced you that something odd is happening with mitochondria inside our cells? I mean, if what I have said is in fact true, and I don't believe I'm lying to you unless people lied to me, okay, then something's up here, gang, right? It's not passing the sniff test, so to speak. All right, so that's the endosymbiotic theory. If you're really motivated, you could certainly Google endosymbiotic theory and find some more information about it. But I just wanted to throw that out there that, at least in the realm of microbiology and how we think life evolved on the planet, there had to be a transition zone between primitive cells of prokaryotic nature and more complicated cells such as the eukaryotes. There must have been a transition. How did that take place? What did that involve? We don't have all the answers, but we have some interesting, tantalizing pieces of evidence which suggest that something weird has been going on over geologically large frames of time. Okay. All right. Okay, gang. I'm going to stop there. Okay, I'm going to stop there. The very last page of your textbook, which in my book is page 102, but somewhere around figure 4.28, somewhere in that vicinity, does give a little bit of discussion about this topic. And so that's why I wanted to bring it up with you. Okay. Do you have any questions you'd like to ask at this time? Okay. So next Tuesday, we will not have the exam. Next Tuesday, I will lecture from chapter 8. If you have questions about the exam you want to ask on Tuesday, I would ask you to hold them until after the lecture, because I don't want to eat lecture time up, but I do want to help you. So as it is on Tuesday afternoon, right after micro lecture, I do have office hours, and I would be happy to help any of you with any of your questions, okay? So we will lecture Tuesday. We will take the first lecture exam on chapters 1, 2, and 4 on Thursday. The lecture exam will take place at 12:30. You will take it during your normal lecture time. So you have to log in to class marker at 12:30 and take the exam, because it will be set up for you to take at that moment of time. So I noticed that many of you successfully logged into class marker. Thanks for doing that. I appreciate it. Are there any concerns, problems, or questions about class marker? It really is straightforward. I mean, you're all computer savvy enough, in many cases more so than I am. It's a pretty reasonable website. So you will take your first lecture, you'll take all your exams on class marker, okay? Any questions about that? Okay. Okay, then thanks for your energy today and your attention. I'm going to stop the recording and move into some conversation about lab stuff now, okay?